When we talk about pregnancy and childbirth, I guess we talk about fertilization a little bit first. And so here is the event of fertilization. And if you remember in the last video, I talked about the layers of cells around the egg that were protecting it. And so you see here those cells. And so it takes a number of sperm to be able to digest their way, to chew their way through those layers of cells. Then they have to release some enzymes that help to start digesting away the very hard shell underneath. But when that happens, they must have a match. So there are little proteins on the head of the sperm and little proteins on the surface of the egg's shell. And not the kind of shell like a chicken egg has, but it functions in much the same way to keep things out. And if those little proteins match up perfectly, then it is recognized as a proper human sperm, and then it's allowed to enter. And here at step 3, it's starting to enter the process, and that starts what's called the fast block to polyspermy. You want to make sure that only one sperm enters the egg, because more than one sperm would then contribute more than one set of chromosomes, and that would be immediately fatal to the baby, or in this case to the developing egg rather, that it would be fatal because it would have more than 2 sets, and that would be a real problem. So what you see there is that little membrane that had those proteins on it to match gets distorted. It's literally twisted out of shape immediately, and that then prevents additional sperm from binding to it and entering. The slow block to polyspermy you see at the last here at step 5. And at that point, basically all the little proteins that would match up with a sperm's proteins fall off of the egg. So the egg is no longer something that recognizes sperm or that sperm would recognize. That would be called the slow block to polyspermy. And between those two, that ensures that one sperm and one sperm only really has a chance to get in. Once that sperm is in, it's not the whole sperm that goes in, it's just really that pronucleus there, that package of DNA, and it enters the egg and then binds or connects up with the nucleus of the egg, and now we have the diploid condition. We have the 2 sets of chromosomes, one from mommy and one from dad. At that point, fertilization has occurred. I just wanted to point out this table here in the book is filled with inaccuracies and assumptions that are occasionally true but not very often. So I just wanted to point out that these events, especially for mama, some of the baby events are off a little bit too, but some of the mama events are quite early in the process. I don't know that 1 week after ovulation you're necessarily going to feel pregnant. Remember, this is 1 week after ovulation. But you maybe have only had an egg implanted in the uterus for maybe a day at that point. So that's perhaps a little bit of an assumption there. And it's going to be quite variable based on individual people. Some females feel something different that is eventually labeled as pregnancy very early in the process, and others might be a month or so into the process before they really feel a whole lot of noticeable changes going on. Suggesting that the menstrual cycle is missed at the 3rd week might be a little bit on the early side as well. And the urine pregnancy test, that's probably pushing the front edge of that as well. So just take a look at this table, or don't, but just keep in mind that some of these things are probably on the front end of possibility, and in some cases, perhaps in many cases, might be a little bit earlier than it would actually happen. So don't take this as the final word, but rather as it could happen this way, but it's probably, again, pushing the envelope a little bit in terms of when it would really happen. What we see here are the steps of the process. So we had at 1 ovulation, then we got fertilized at 2, and then what you have within about 24 hours after ovulation is you're in step 3, which is cleavage. So what's happening there is this single cell that was an egg, bonded or bound with the nucleus of a single cell of a sperm, and it's still now a single cell, but then it starts dividing about probably 20 hours or so after ovulation, assuming it was fertilized. And for the next couple of days, we said it was about a 6-day trip down the fallopian tubes. For that time frame it's dividing, and by the time it gets down to the uterus, it's gone from one cell to a couple of hundred cells, and they're getting smaller and smaller as you go. Then they're at step 5 and step 6. It's a sort of a hollow ball of cells that then implants into the wall of the uterus. It's implanting into the uterus so that it can get a blood supply, because it needs nutrients far more than it had stored in the egg. And what we see here is an early placenta. So a placenta is basically just a series of blood vessels interacting between mama and baby, in which baby can pick up nutrients and oxygen from mama's blood and drop off waste products into mama's blood. But mama's blood doesn't actually enter the baby and doesn't actually even register the baby's presence in terms of the immune system response. So it's a neutral interface. Mama dumps in blood, baby takes what it needs, and then mama takes the blood back. And at this point, this baby is several weeks into the development process. So it's probably a little bit inaccurate to suggest that there's still much of a yolk sac left. But the yolk sac basically was the energy stored inside the egg that was available for that baby's development. You're familiar with the chicken egg, and if you crack it open, it has that yellow yolk inside. And that was what was intended to be the energy source for the baby chicken as it grew inside the egg. Now in a chicken, it had to have a pretty good size yolk, because it needed to have all the energy it needed to completely grow and develop up until the point of hatching inside the egg with no additional input from mama. Because the egg is laid outside of the mama's body, there's no chance to replenish the nutrients. So the yolk sac in a human egg doesn't need to be that big, because it's only there really to get you through the first, let's say, week and a half to 2 weeks of that little ball of cells existence. And by the time you have recognizable pieces and parts here in human appearance going on, there's a solid placenta, which is then just a renewable energy source for the baby as it grows. So early in that process, up here at the 18-day mark, they've labeled, it was implanted, but not a whole lot of placenta there. Then we see more and more placenta around the edges as it goes, and we're seeing some major changes inside. So from day 18 to day 35-ish, it goes from a kind of a mass of cells into something that definitely has a head end and a tail end and some development along the way. So here at the bottom, we can see here's the head end down here, and then here's the tail end. We've got the umbilical cord here that's formed, and that's basically the connection between the baby and the placenta. And so really here, the first thing that's developing in terms of deciding what's what is the nervous system. And this is why it's so incredibly important to avoid alcohol at any point in which a female might be pregnant or might be getting pregnant. Because in this very early stage, certainly before you would necessarily know that you're pregnant in terms of feeling or having missed a menstrual cycle or anything else, the baby's nervous system is developing. And so the brain is starting to take form, and the spinal cord. And so if alcohol is present in those early stages of development, it can greatly interfere with the development of the nervous system, really resulting in a less than perfectly functioning and somewhat deficient nervous system, and that's called fetal alcohol syndrome. That can range from a very mild case of intellectual impairment that may not be necessarily noticeable, all the way up to a very severe case of intellectual impairment, along with some significant physical deformities of the face, head, and body. And so that would be a problem that you can directly point a finger at mama and say, it's your fault. You did this and you caused this problem. But because it's preventable, we don't need to have anybody suffer from that. If there's any possibility of being pregnant or getting pregnant, just don't consume alcohol, and then you don't have to worry about it. If we look at that ball of cells, before it starts taking shape in the body form, it's just sort of a ball of cells, and they have colored them here as blue, pink, and yellow. And you can then trace those cells to what part of the body they're actually going to become. So as a ball of cells, The cells have already been told, here's the kind of tissue that you're supposed to go form. And a cell is not necessarily told, you go form a finger. But it's told, here is what you have the options to become. Go do something like that, and that exactly which of those options it takes will be later determined by hormone levels. But early in the process, it's told, here is the list of things that you can become. And so at this point, we're no longer going to see very much in the way of embryonic stem cells. Rather, we're going to start seeing more along the lines of adult stem cells. So embryonic stem cells are those that have the ability to become anything that the genetic code could possibly code for. So it could become any kind of tissue in the body at all. An adult stem cell has gone through this process of determination in which it said, okay, here's your range of possibilities. You can no longer become anything. You can only become A, B, C, D, or E. And so those stem cells are useful in the world of stem cells, but not as useful as embryonic stem cells because their options have been limited at that point. Certain genes have been turned on, certain genes have been turned off, and for the rest of their development, they're going to stay that way. Here is a human baby. At this point, we're a couple of weeks into the process. This baby overall is probably somewhere around 1/8 of an inch long. So it's very, very tiny, but we can see here limb buds. So we've got some developing arms here. We've got developing legs here. But notice we have a tail. And so that tail will sort of fuse or absorb into the rest of the body so that by the time the baby's born, you probably won't see a tail anymore. But your tailbone is a remnant, a detectable remnant of that tail that you had early in the process. Also, the pharyngeal pouches up here were precursors to gills. So in a way, human babies before birth, early in this process, had gills and they had a tail, neither of which are visible at birth, but they were present. If you were to look at a chicken or a elephant or a fish or anything else really at this point, they're going to appear quite similar at this stage of development. And so you can say then that that gets into some evolutionary arguments perhaps, but most higher level vertebrates look very, very similar at this stage of development because they're all really following the same body plan. The only difference is what direction do these pieces take once they've achieved the status of existing. Do they become arms? Do they become fins? Do they become long legs? Do they become something else like that? Does the tail stay and have a function? Does the tail stay and not have much function, or does the tail disappear altogether? So those are all things that become different after this stage of development. One thing that happens in the baby, and really the first functioning organ, we said the first developed organ is the brain and spinal cord, but the first functioning organ is the heart, and that comes along a week or two after the brain and spinal cord get going well with development. So in this particular case here, this is a pretty well developed baby, and we can look at the heart. And what we see is the heart actually has some openings between the chambers that it wouldn't normally have as an adult. That's normal in an unborn baby. And it actually explains why newborn babies sleep so much, and we'll get to that in a little bit. But basically, a lot of that blood skips going to the lungs altogether and goes from the right side of the heart, has oxygen pour, to the left side of the heart and straight back out to the body. Because the baby's not breathing at this point, there's nothing in the lungs to do gas exchange with, there's absolutely no point in sending a lot of blood there. All the oxygen is being picked up from the placenta, and that's coming in through the umbilical cord. And you can see here the oxygen-rich and oxygen-poor parts of the cycle. And what we'll see is that at this point there are certain connections of blood vessels. Some of those connections associated with the umbilical cord will completely disappear after birth because they're no longer needed, so they're just reabsorbed by the body. And others will take on a little bit different functionalities and purposes after the baby's born. There are a couple of options here for testing the baby genetically to see if there are any underlying genetic problems or potential problems maybe that might be there, and that would then allow you to make a better informed set of decisions about what to do with the pregnancy if there's a potential problem there. Probably for the average pregnancy this is not really necessary, but if there are particular genetic problems in a family, then perhaps this testing might be a little bit more important. Basically, it tells you what the likelihood is of a particular problem or not existing. And none of this is 100% guaranteed, but it's pretty reliable in the grand scheme of things. As good as we can really hope for it to be, probably. And so if you detect a problem early in the process, you can perhaps do a little something to make it a lesser a problem. You can perhaps try to fix the problem, or you can choose to perhaps terminate the pregnancy at that point. So the first one here is amniocentesis. In amniocentesis, they're going to go in with a syringe and draw out some amniotic fluid, which is going to contain some fetal cells, and then test that. And the good news is the chances of harming the baby during this test are only about 1 to 2%. So it's really a pretty small percentage. I personally would be comfortable with that sort of risk. The downside is the timing of it typically occurs around weeks, let's say 14 through 20. And then the results take around 5 weeks to get back. So if you did it at 20 weeks, you get the results back at week 25. You have the doctor's appointment to have the results explained to you at week 26. And at that point, termination of the pregnancy in a fair number of states is no longer a legal option. So the type of genetic test that you do really determines when you can find out what the results are, and that really determines what your legal options are at that point. Some states now have extended the option for termination of pregnancy. Some of you like to call it abortion, and that's okay. Some states have extended that out to practically right before the baby comes out on its own. And there are a number of ethical concerns with that, and personal beliefs and things like that come into play as well. So it gets very, very complicated. So scientifically speaking, typically there's not much need from a medical standpoint to look at termination of a pregnancy at a very late stage like that. So, biologically it's not of overly great use to have a late-term pregnancy for the most part. The second option here is chorionic villi sampling. The downside here is it takes the risk to the baby and doubles it from 1 to 2% to somewhere around 2 to 4%, which is still single-digit risk, so it's not incredibly risky in the grand scheme of things, but the risk did go up. What they're going to do there is they're going to actually suck off some of the placental tissue and test that. The good news here is that you get the results typically within 24 hours, and it can be done somewhere around weeks 10 to 12, maybe 13 or 14. So you can do it earlier in the process, you get results faster, so then your options are certainly much more in the way of options available to you in an earlier stage of pregnancy. The third possibility here is looking at, and this is kind of a new idea, pre-implantation genetic diagnosis, basically looking at the eggs in the ovary to see what kinds of genes are present in the ovary, and then that would tell you what kinds of possibilities you might get in the world of eggs. I've not seen a whole lot of information about that one, so it's perhaps not incredibly useful because it can't give you a sample of all the eggs. And remember that all the eggs and all the sperm are genetically unique. So what one egg may be perfectly fine, another egg right next to it might have some serious problems. So this one's going to be kind of limited. Whereas the amniocentesis and the chorionic villi sampling, or CVS, gives you results about that baby, not a potential baby out of this mix, but that baby may or may not be represented in the test. So probably the CVS and the amniocentesis would be more useful in determining what the issues are on this one, not necessarily what the gene pool contains. My 2 cents for what it's worth. Early in that process, so up until the first couple of weeks, you can look at that baby with any kind of ultrasound, any kind of imaging or scanning technology, and it's going to look exactly the same no matter which gender it's going to be. Or we really should say no matter which gender it is. Gender is quite simple. Society has made gender very complicated these days, but it's really quite simple. The presence of a Y chromosome equals male. The absence of a Y chromosome equals female. No matter what's expressed, no matter what develops or shows, no matter what a person identifies as or is oriented to, gender is a genetic thing that is a Y chromosome present male, Y chromosome absent female, end of story. So scientifically, biologically, gender is quite simple. But what you see here is you see gonads, you see the kidneys drawn in here, but you also see a pro-bladder device which can become bladder or not bladder, but we also have Wolffian ducts and Mullerian ducts. If the individual is a male, the Wolffian duct will become epididymis and vas deferens and connect up with the testes directly. If it becomes female, the Mullerian duct will become fallopian tube. But it's equipped with both, so no matter what the expression is, both pieces and parts are present. It's just a matter of which one develops and functions and which one disintegrates or becomes something different. So we can follow through here the SRY present. And SRY is a gene that's on the Y chromosome. So this goes back to Y chromosome present, you get male. The only question is, what kind of visible male do you get? Does the individual appear? And the book goes into this in a little detail. Does the individual appear male or do they appear female even though they're genetically male? That has to do with how well the testes function and not necessarily with anything else in this process. But here it becomes again, the epididymis vas deferens, and then eventually descends into the scrotum and the tubing follows behind. If it's the female, so there wasn't an SRY gene present, we don't get high levels of testosterone, and the gonads become ovaries instead of testicles. The Wolffian duct kind of disintegrates and becomes connective ligaments and tissue and not anything actually physiologically functioning. And then the Mullerian duct does the fallopian tube business. And in the case of the SRY gene present on the male, the Mullerian duct pretty much disappears, might serve a little bit of connective tissue function, but really is not anatomically detectable after development. So again, these started out exactly the same and differed based on a gene that was there or not there, and then it did things or didn't do things based on its presence. Typically, for the average individual, whatever they are genetically is what they very obviously appear at development. And again, if they don't appear that way, that's a hormone imbalance. And that had to do with the testicle or the ovary either doing what it was supposed to do or not doing what it was supposed to do. But underlying the gender is what it is, no matter how you look at it. If we look at external genitalia in their development, again, everything starts out the same. So at that 6-week mark, there was a urogenital groove, there was a genital tubercle, that little lump at the top there, and then there was the folds of tissue on the side here. If we follow that through the SRY present, so it's going male with testosterone, that genital tubercle there becomes a penis, and along with that groove, these folds of tissue at the side become the scrotum. If that gene was absent, it's going the female route, this genital tubercle becomes a clitoris. Those folds to the side, or rather the urogenital groove becomes vagina, and the folds to the side become the labia. So the same pieces and parts are there again. The difference is, are the hormones present from this gene or not, which then determines which direction it goes. So just like we saw with the Wolffian duct and the Mullerian duct for the fallopian tubes or the vas deferens, the external genitalia work the same way. It either goes one way for a male or one way for a female based on those hormones. And this is where that ambiguous gender concept comes in. Was there enough hormone to clearly push the expression of these external genitalia one way or the other or not? and then that can lead to some issues there. But again, underlying this really is the gene. If the gene's there for the Y chromosome and for male, or it's not there for a female, and that's really as simple and as complex both at the same time as it needs to be. We could go on and on and on about the pregnancy process and what it does to the body and what's involved in that process. Really, we could spend an entire class talking about this process, from sperm and egg production to fertilization to pregnancy to childbirth and everything in between. And I actually had a class like that in grad school, and the entire semester was spent on this idea of what it takes to reproduce. And so it was a very interesting topic. And again, we could go on forever with it, but hopefully you learned a lot and this all made sense. There's, again, a lot of possible controversy in there with gender expression and gender identity and orientation and all kinds of other things there. And that's perhaps best left to a psychology course, because we really don't have any explanations or answers for those issues in the world of biology and science. So if we can't deal with it, we leave it for someone else to deal with, and that's where that issue falls. So again, hope this all made sense. Thanks for joining us. We'll see you next time.